All right, I think we can go ahead and get started now. Hello everyone, and welcome to day 3 and our last day of our flow cytometry series. So today we have Dr. Sharmila Biswas, who is joining us to share some best practices when running flow cytometry for specifically phosphorylated proteins. But before I pass it off to Sharmila, I just want to share a few company slides to introduce our company, Protein Tech, to you. So, Protein Tech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass many different research areas. We also offer immunoassays such as ELISA kits and IHC kits, as well as cell separation kits and antibody labeling kits. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors. And all our proteins are expressed in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins. And that ultimately increases their bioactivity compared to proteins that are expressed in, for example, E. coli systems. We also have our Chromatec branded nanobody-based reagents. So nanobodies are the antigen binding domain of heavy chain only antibodies, and they're about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity. And this makes them ideal for several applications, including immunoprecipitation, immunofluorescence, and protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And Protein Tech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. So, as a company that is driven by scientists, we're here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. We also know the importance of product validation, and we offer open access validation data. When we're testing new products, we do validate with relevant sample types, appropriate experimental conditions, and using knockout, knockdown models whenever possible. Also, as antibody experts, we do have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform that we use to manufacture our recombinant antibodies. Also, with 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world so that you can get started on your experiments right away. As a company, we really do measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. And so this graph over here shows that over our company history, our products are being featured in more and more publications each year. And as of now, we have over 300,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also really quickly want to highlight that as a company, we strive to make the searchability of our products on our website as user friendly and informative as possible. So, we've recently launched some new updates on our website to help our customers select the right antibody. This includes our unique 3D epitope viewer that allows you to interact with epitope structures and explore antibody binding sites so that you know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need. So we have our immunogen region selector toggle, and that allows you to filter for antibodies that are generated from your region of interest. Additionally, you no longer have to filter through all the variations of an antibody from the search results. You can view all formats of an antibody, including conjugations as well as buffer formulations, on just one page, which ultimately is going to streamline your process for antibody selection. So, with that, I would like to go ahead and introduce our speaker for today, Dr. Sharmila Biswas. So, Sharmila is a senior scientist in the Dissolution Science Department at Johnson & Johnson Innovative Medicine in Belgium. With 8 years of experience in cancer systems biology, her expertise includes flow cytometry, molecular biology experiments, and kinetic mathematical modeling. As a postdoc at Systems Biology Ireland, which is associated with University College Dublin, she utilized CRISPR-Cas9 gene editing technology to identify epigenetic vulnerabilities in childhood leukemia. Her PhD research at the Indian Institute of Technology, Bombay, focused on the dynamic crosstalk regulating TNF alpha signaling induced apoptosis through a systems biology approach. During her PhD, she served as the flow cytometer operator responsible for running samples from multiple departments at IIT Bombay. She independently conducted and analyzed flow cytometry experiments using FlowJo software and played a key role in organizing and speaking at annual flow cytometry workshops. She has published multiple research articles in international journals, including 3 first author and 1 second author publication. Additionally, she has actively participated in various public engagement initiatives in Dublin. Her research has been presented at over 20 national and international conferences, earning several awards for best oral and poster presentations, as well as travel grants. So with that, I'd like to go ahead and pass it off to Sharmila. Thank you so much for being here. I know it is a Friday evening for you, so we really appreciate you taking part of your weekend to come and talk to us.
Okay, am I audible? Yeah.
Yep, yep, I
Yeah.
can hear you.
Okay. So thank you, Afrida, and yeah, I really appreciate you like doing all the different. You were very kind to invite me on this platform and also answer all my questions regarding the workshop. So thank you for that. And yeah, I would also like to thank all the participants who are tuning in from all across the world. Based on your timing, yeah, I also appreciate your time coming here. So yeah, a very warm welcome to everyone, and I would say maybe good morning, good evening, or good afternoon from wherever you are tuning in. And welcome to the final day of the flow cytometry workshop series. So before diving in, I would first share my screen. Uh, Yeah, could you just let me know if it's visible?
I see your screen, but not as like a presentation yet.
Okay. Is it now
Yep,
in the
perfect.
presentation mode? Yeah, okay.
Yes.
Okay, so yeah, today I'll be talking about the Phospho-Flow Cytometry, which we use to explore the cell signaling pathways. So in today's talk, I will be covering these following topics. And I know that many of you might be very well aware of what flow cytometry is and all the basics about the flow cytometry and the flow cytometer. But yeah, just to cover the entire audience that's present here, I would just give a brief introduction about what flow cytometry is. So the flow cytometry name, as it suggests, if you break it down, it's a flow, which is fluid, cyto is the cell, and metry is basically the measurement. So flow cytometry is basically, I'm sorry. Yeah, it's basically measuring the properties of cell when they are flowing. So this is basically like there are single cells in suspension that pass a laser beam which produce the characteristics light signals which are analyzed by different detectors. So based on the functionality, the flow cytometry can be divided into two main categories, that is the cell analysis and the cell sorting. So in cell analysis, we measure the different characteristics of the cell, such as based on the cell size, the complexity or the granularity, and also the different fluorescent intensities by the fluorescent markers which are attached. And the cell sorting is sorting these cells based on these characteristics. So the basic design of a flow cytometer consists of three main components. First is the fluidic system, which brings the cells to the laser interrogation point. Second is your optic systems, which collects the light scattering from the cells and also the fluorescent emission. And finally, it's the electronic system which converts this light into a digital, from the analog to the digital conversion, so that you can analyze it with your computer. So moving ahead, the first part, which is the fluidics, it's basically which delivers cells and aligns them in a single file to ensure that they pass through the laser interrogation point for analysis. The main heart of the fluidics is the flow cell, which are designed to accelerate and space out, focus, and basically align these particles in a single file. And this is done by hydrodynamic focusing, which uses these fluid dynamics to ensure that your cells pass through the laser beam one at a time, and which improves your data accuracy and allow for the single cell analysis. The second part as the optics, where you analyze the cells and particles by using lasers to illuminate them and also then detect the light scattered and the fluorescent signals which are emitted. So there are four main components of the optics. First is the different lasers which are used as a light source which illuminate your cells or samples. And then there are lenses which focus this which focus the laser to the sample stream. And then there are optical filters which include the dichroic mirrors and the bandpass filters, which basically splits the different lights. So when a cell is illuminated with light, it scatters signal in all different directions. So you need something to split up these wavelengths at split up it into specific wavelengths so that you can detect the different fluorochromes, so which is done by the mirrors, filters, and the detectors. So how optics basically work in flow cytometry is the first is the light scattering. So when a cell passes through this laser beam, it scatters light in different directions. And the two information-rich scatter components that we get is the forward scatter, which measures light along the incoming light, which is the diffracted light, and it is proportional to the cell size or the cell surface area. And the second one is the side scatter, which measures the light coming in the 90-degree angle, which is the reflected and the refracted light, and it is proportional to the cellular complexity and the granularity. The second is the fluorescence. So the fluorochromes which are present, they basically absorb the light energy from the laser and at a given wavelength, and then they emit a portion of this energy at longer wavelengths, and these two processes is called the fluorescence excitation and emission. So what basically happens is when your fluorochrome is resting at the ground state and it is illuminated by a laser, it absorbs the photons from the laser, and then the electrons in your fluorochrome gets excited and it moves to the excited state, and this is the fluorescence excitation. And it stays in the excited state for a very few nanoseconds, and with the change in the conformation of your fluorophore, a part of the energy is emitted as heat, and then the rest of the energy it jumps down to its ground or the resting state with a lower energy level. So as the emission is a very low energy, so it has a higher wavelength as compared to the excitation. Always the spectra will have a and this is important to understand, this is important to consider for how good your fluorochrome is, because large Stokes shift corresponds to easy fluorescence detection. So most of the flow cytometry platforms have cytometers which are equipped with some or several of these lasers, which I have depicted here. And this laser, when they illuminate your cells, there is the light is scattered in various different directions, and then you need these optical filters which basically confine these cells based on a specific wavelength or a range of wavelength, so that you can separate out the different fluorochromes based on the different wavelengths or the different emission spectra. And this is done with the help of the filters. So you basically have three different filters. That is the long pass filter, which, as the name suggests, it will allow the passage of the wavelengths which are longer than, for in this case, it's 750. So all the wavelengths which are higher than the 750 nanometer, it will allow to pass through. And then there are the short pass filters, which is that all the wavelength which are less than 500, it will allow it pass through, the others will get reflected. And the final is the band pass filter, which operates in a range that is here, the band pass filter 525 plus or minus 25 nanometers. So anything in which falls under this wavelength range, it will allow it to pass through. So basically, one tip here, which I want to share with everyone, that before starting any of your flow cytometry experiment, please know about your instrument, or just talk to your flow cytometer operator of what are the lasers and the filters available before even thinking of planning to do an experiment. So the next thing I want to talk to you about is the spectra viewer. So now, as you know, that there is this excitation and the emission spectra. So what is this used for? Is the excitation spectra is used to define the laser line, that is the best laser line that you can use for excitation, and the emission spectra is used to select the best channel of the optical filters which will capture this emitted wavelength. So here we can take the example of FITC. So if you see the excitation spectra of FITC, which is depicted by this blue dotted line, the maximum excitation of FITC is around 496 nanometers, and this falls in the blue spectrum range. So this means that it's the blue laser that will cause the maximum excitation or the absorption of this fluorophore. And the emission spectra, which is depicted by the pink histogram here, it ranges from around 475 to 610 nanometers, and the maximum emission is at around 525 nanometer, which falls in the bandpass filter of 530 plus or minus 15. So this means that it falls in the green filter. So that's why your FITC appears green. So basically the fluorescence color that you get is usually, it refers to the color of the light of the fluorochrome, which it emits with the highest probability. And a few points to note here, that you don't need, or you won't always be able to perfectly match the fluorescence absorption maxima with the different lasers which are available, but you have to try to get close enough to guarantee that your fluorophores is excited to the best possible way. So for example, let's say this is the PE fluorophore, and the PE fluorophore can be excited by both the blue laser, which is 488 nanometer, and the yellow-green laser, which is the 561 nanometer. But the excitation efficiency differs based on the maximum excitation for this fluorochrome, which is around 575. So if you see, for the 488, it excites around 40%, while for the 561 laser, it excites with 76% efficiency. So ideally, because there is, with 561 laser, there is more excitation. So ideally, you will get more emission spectrum. So this means that when you are working with antigens which are very dim, so you should ideally use the 561 laser, so to get a brighter signal as compared to the 488 nanometer laser. So with this, I will now jump to the actual Phospho-Flow cytometry that everyone is excited about. So what is Phospho-Flow? So Phospho-Flow is basically a flow cytometry based technique which is used to measure the protein phosphorylation at the single cell level. So protein phosphorylation basically is an indicate of the activation status of the signaling proteins, that if the signal is in its active form or not. So if it is, most of the cases, if it is phosphorylated, it is an active protein, and if it is dephosphorylated, it is not an active protein. So this provides insight into the various signaling pathways, and also to understand the various cellular activities. So if you have to measure multiple signaling pathway given a particular antigen, and also want to check how the cells are behaving with changes in the signaling pathway, then Phospho-Flow is the way to go. So there are various advantages of phosphoflow cytometry. The first being, of course, the single cell analysis because it collects data at individual level. So there are conventional techniques like the Western blot, but the Western blot actually gives a more combination effect, that is all or none response, as compared to the flow cytometer, where let's say if you have you think that you have a homogeneous population of cells, and when you do a Western blot, you get a negative or a positive response. There is no intermediary response, or you know that yeah, some of the cells might be the population might be heterogeneous in nature, or if it is homogeneous in nature. So with flow cytometry, you can see that here some of the cells, they have more intensity, or they are expressing the fluorophore more as compared to some of the other cells. So this means that the protein expression is basically a bimodal protein expression, that not all the cells are expressing the protein in the same intensity. You have some cells which are expressing at a low intensity, and there are some cells which are expressing it at a much higher intensity. So because of this bimodality, also there are some cells which you see, so now people are more working towards the precision medicine, because now there are some group of cells which behave very differently than the population, and then these cells actually then turn to they cause this resistance in cancer treatment and everything. So with flow cytometry, you can identify if there is difference in the signaling or properties of the two different groups of cells or populations which you have identified with flow cytometry. And apart from this, there are also rare subsets which can be identified. Like in this case, there's only one cell which has a very high expression level as compared to the rest of the cells. So it's just like a 1% chance to detect this, which can be easily detected with flow cytometry, but there is no chance to detect such kind of expression level with Western blot. So also based on the shape of your curve or your histogram. So if it is a nice Gaussian distribution, then you know, yeah, all the mostly all the proteins are or all the proteins are expressing at a very normal way. But if there is a skewedness towards the right or the left, then there is some population of cell which is behaving very differently than the rest of the population. And also with Phospho-Flow, you can do multi-parameter analysis, because you have these different fluorophores with the different excitation and emission range, and then you can select yeah, different fluorophores, and you can measure these parameters simultaneously on each individual cell. And this is very helpful for measuring the immune cell signaling, where there are multiple markers and also the different signaling events that you want to measure. It's also possible to measure the intracellular levels or the surface protein markers at one go. The next thing is this protocols of phosphoflow cytometry is pretty rapid, and you can easily perform them in a 96 well plate in parallel. So that you can run in one go 96 samples, and it's easy to visualize and also compare these samples among the different experimental conditions and across different cell lines. So with the histogram, it's very easy to do a comparative analysis if there is a bimodal expression, or if there is a skewedness towards one of the side, either the right or the left side, and or if there is a smooth histogram.
Hey,
And
Sharmila, I'm so
yeah,
sorry to interrupt.
no
So it
problem.
seems like there's a few people who are having difficulty hearing you. They're saying it's getting a little
Okay.
broken. I don't know if maybe is the headphone or speakers, or sometimes I
Yeah,
know like if
maybe.
my hair gets caught on the mouthpiece,
Uh, maybe that
that
is the
kind of
case. Yeah.
thing.
Uh, just I will then. Is it better now?
That's much better. Yeah.
Okay.
Sorry about that.
No problem. Yeah, thanks for letting me know. Yeah, so
It's starting to break again. I don't know
okay.
why. Yeah, because we tested it out too, and it had sounded fine. But yeah, I'm hearing it a little
Uh,
broken again.
okay. Yeah, let me see what I can do now. Should I maybe?
Now I can't hear you.
Hello, is it better now?
Yes, now it's better.
I hope
So
it stays
hopefully,
like this.
yeah, hopefully.
Uh,
Okay.
okay. Sorry about that. Yeah. So yeah. The next thing is the statistical significance, where you, because it's the data is very rich, where you will get the different properties such as the population mean, median, the standard deviation. So yeah, you can do a lot of things with the rich data. So it's a statistical significance, which is not the case that you get in Western blot or ELISA. So with that, we will now talk about how to do the phospho or how to proceed with the phosphoflow staining methods. So there are three main or major important steps that you need to focus on when you are performing a phosphoflow staining. So the first is the fixation. As the you have to capture the phosphorylation status, so the cells needs to be rapidly fixed in order to keep the original phosphorylation status of the target proteins. So the fixation step is very crucial to capture the exact phosphorylation, and also if it is a time dependent experiment, then you need to make sure that uh, the time that you are planning to measure, it's the exact time that you also fix the cells. And the next is the permeabilization, where the specific antibodies must be able to get access to their target epitopes. So effective permeabilization is also crucial, and followed by the antibody staining, which will then stain your phosphorylated moieties, which recognize and stably bind to your epitopes. So uh, Krutzik and Nolan in 2003, they basically talked, so they were the first group to come up with intracellular staining experiments for phosphoflow. So they basically compared different methods, and they developed the first effective and reproducible approach for the intracellular phosphoprotein detection using flow cytometry. So briefly, you take a biological sample and then you stimulate it with a cytokine or a small molecule. Then you apply these cells through a cross-linking agents or fixating agents, that is formaldehyde, followed by the permeabilization with detergents or alcohol. And finally, you stain the cells with phosphospecific antibodies that have been conjugated to different fluorophores. And finally, the cells are analyzed by flow cytometry to determine the level of phosphorylation in your stimulated and the unstimulated groups. So the fold change of in the median fluorescent intensity between the stimulated and the unstimulated cell revealed that the fixation with formaldehyde and the permeabilization with methanol was the best combination, and it was confirmed in multiple signaling pathways that these two combinations worked really well. The experiment suggested that the concentration of formaldehyde and also the time and temperature of incubation might result in changes in the phosphoprotein staining. So it's important to check if the formaldehyde concentration that you are using, and also the incubation time that you are performing is actually effective for the phosphoproteins that you are planning to measure. And this is also you also check that the stability of the phosphoepitopes is hampered if you measure the levels of the phosphoprotein immediately, or if you preserve it overnight in cleaning buffer. So ideally 70 to 90% of the phosphoepitopes remain stable after fixation and permeabilization. But yeah, I would say yeah, you should perform the experiment as early as possible as you have fixed or permeabilized your cell, so that there is less variation of the levels of the phosphoproteins. And now I will discuss a few technical considerations for the phosphorylation state analysis. So the first is the antigen accessibility. Now, unlike typical protein epitopes, the phospho-specific epitopes are often buried by protein-protein interactions. And after fixing the cells with cross-linking reagents such as formaldehyde, these epitopes may be buried within the protein interfaces. So like experimental evidence have suggested that with proper permeabilization, and specifically with alcohols, for example, the methanol, most antigens can be measured efficiently. So the permeabilization step is as crucial as the fixation step, so that your antibody gets access to the antigen. The second is the cellular localization. So the location of the epitope within the cell may limit its accessibility. So basically, like there are nuclear antigens such as the STAT transcription factors and phosphorylated MAP kinases like ERK, the JNK, the P38, AKT, so many are there, and you may require different fixation and permeabilization techniques than antigens which are actually present in the cytosol or at the plasma membrane, such as the PLC gamma or the ZAP70. So various protocols has to be screened for different classes of phosphoepitopes and protein families. The third is the stability of the phosphoepitope. The rapid and efficient fixation techniques are required to prevent the dephosphorylation or further phosphorylation. Because of the transient nature of the intracellular signaling events, the fixation techniques which are used for phosphospecific analysis must be rapid and efficient at freezing this signaling to prevent the dephosphorylation or further phosphorylation. And along with this, the stability is also of particular concern, because when you are applying the technique to patient samples, let's say, which are already cryopreserved because of the effects of the freezing on the both surface and intracellular epitope, the stability of the phosphoepitopes do change. So you should keep in mind when you are dealing with patient samples. And the next is the antibody selection, which is a very crucial thing, because yeah, it can give you false positive results if your antibody is not able to bind to your antigen. So selecting and screening of antibodies to find those that stain the antigen of interest most efficiently and specifically is very important. Also, there are many antibodies that work extremely well for Western blotting, but they do not perform as well when you put in the context of this fixed and permeabilized cells during flow cytometric analysis. Apart from this, there are also different antibody clones, which against the same peptide immunogen can show disparate staining levels. So yeah, you should keep in mind before taking different batches of antibody clones. You always make sure that the batch number is same for the antibody that you're using. So therefore, one must use caution when attempting to stain the phosphoepitopes for flow cytometric analysis, because sometimes you might get negative results which may not represent actually the lack of phosphorylation, but rather the lack of epitope availability and therefore lack of antibody staining. So also I would like to highlight that in this context, a positive control is very important here, because a positive control will make sure that stimulation conditions, let's say for example, the PMA, which is used for ERK, you get a positive signal to know that your experiment or your protocol has worked, and maybe the antibody that you are using for measuring the ERK is not getting access to the antigen for some reason. So you can have a check by using a positive control. So yes, that's very important for the antibody selection. And the next is the fluorochrome selection. So there are various fluorophores which are available, and some fluorophores are smaller in size, such as the FITC, the Alexa 488, and the Alexa 647, which provides the best staining characteristics. But there are also large protein fluorophores like the PE or the APC, which are very slow, which allow slow antibody entry, and it affects its binding characteristics. So you should be mindful when selecting the fluorophore for your phosphoprotein analysis. So now I would be talking a bit about one of the applications of Phospho-Flow cytometry, that is the fluorescent cell barcoding, which was basically like it was a technique which was introduced to help in better managing your multiple samples, or in doing a high throughput flow cytometry analysis with the intracellular staining. So the fluorescent cell barcoding is basically you provide a unique signature or code to your samples. It basically facilitates the simultaneous analysis of samples by pre-labeling the different samples with different intensities of various fluorophores before the antibody staining. So here, if you see, like I have 4 different samples, and I can add different concentrations of one FCB dye, or the fluorescent barcoding dye. And these dyes are basically amine bound dyes. So it goes and binds to your amine residues of the protein. So this is a covalent bound. And then you can wash away the unbound dyes. And finally, in one combination, in one tube, which is the combination tube or the combo tube as we call it, you can add in all the 4 samples which are barcoded with the different concentrations of the dye. So now you can backtrack of which sample belongs to, based on the concentration of the dye, which sample belongs to which of the dye concentration. So here with different dye concentration, so you used 4 different dye concentrations, so you can see a perfect separation of all the 4 dye concentration in the different dyes that was used. And also when analyzing by flow cytometry, each sample can be differentiated according to this pre-labeled fluorophore. So there are various advantages of this, like you can do simultaneous analysis of multiple samples, and then there is a rapid detection of markers, and it also involves in uniform staining, because now you are adding all the 4 samples into one combo tube, and so the variability in staining or pipetting or handling errors, it also diminishes. And of course, it minimizes the antibody cost by a great deal, because this is one of the dye barcoding that I'm talking about. I'll also talk about 2D barcoding, where you can combine 2 different dyes and also analyze more than 4 samples in one tube. So the 1D barcoding, which I have already talked about, or the 1-dye barcoding, which is called the 1D barcoding, the main challenge is you need to get separate clusters so that you can deconvolute them later on. So this is a challenge while doing barcoding, but with efficient protocol so optimization, you can easily achieve this. And there is also 2D barcoding, which is the 2-dye barcoding, where instead of 1 dye, you can use 2 dye, 4 different concentrations of each of the dye. And if you combine them, and if you make a combination of the different dye combinations, so you can get 16 samples in one combo tube. So this means that for each sample is coded with a different barcode, like the different concentrations of the dye, and you know that which sample corresponds to which particular sample treatment. So once we have standardized our FCV protocol, now we want to combine it with the intracellular staining, because until now I was not adding any antibody to it. Now to measure the various phosphoprotein levels, I want to add the antibody in the final combination tube. So this is one example of experiment where you want to measure, let's say, the TNF signaling at different time points over a 24-hour time period for different proteins. So in this case, you can use the FCB where the different time points can be barcoded with different concentration of the dye. And then in one tube, which corresponds to, let's say, 4 different time points, you can then add the antibody instead of adding it individually to each of the tube. And finally, by analyzing your flow cytometry, you will get the 4 different clusters which corresponds to the 4 different dye concentrations, and then you can deconvolute it to get your median fluorescent intensities of the different phosphoproteins. And based on the different fluorophores, uh, each cluster will have information of 2 or depending on the number of fluorophores you have used, the fluorophore will give information on the phosphorylation status of the different proteins. So what deconvolution method exactly is, so I will talk about it in more detail here. So let's say this is your experimental plan or the experimental design where you want to measure the phosphoprotein level over a 60-minute time point and in replicates. So you can use the 2D barcoding where you use the 2 dye, the Alexa 350 dye, which corresponds to the different time point. So each time point is corresponding to the different concentration of the dye. And with the 647 dye, it can correspond to the different replicates. So if you combine these 8 samples into one tube, it will be your one combo tube, and then this will be the second combo tube. So if you run one of the combo tube, you will get a similar cluster where all the 2 different rows corresponds to the 2 different replicates, and each cluster here corresponds to each time point. So this means that with only one sample, by running only one sample, you have generated the data for almost 8 samples. And this also significantly reduces the cost of your antibody. So the deconvolution is now you want to extract data out of the phosphoprotein levels out of these clusters. So when you plot the scatter plot, if you plot the DAPI, which is the filter that we used for Alexa 350 with respect to the side scatter, you get the 4 different clusters, which is corresponding to the 4 different time points. And now for each cluster, each time point, if you again select this and then look into the another filter, that is the APC, which is corresponding to the Alexa 647 dye, you will get 2 2 clusters, which corresponds to the 2 different replicates. So now for each replicate, then you will get, you can generate the entire signaling profiles for, and each replicate, it's actually in one experiment, you have actually captured the entire thing. So this is pretty cool. And one thing which I would like to talk about here is, because I'm now talking about 2 or more than 2 fluorophores, so automatically the term compensation comes here. When you're using 2 or more fluorochromes in combinations, the chances are usually high that the emission spectra will have an overlap, or there will be an overlap between the 2 emission spectra that you are using. So the overlap, I mean by overlap is, if you see here, there is a one detector of FITC and there is another detector of PE, and you see that around 7% of the signal is spilling into the PE detector. So this indicates that some percentage of the cells, some percentage of your cells are actually showing a PE signal, which is actually not the case. So if you see such kind of data in the PE channel, you will get a positive response, but that is a false positive. So basically, you need to compensate, or you need to get rid of this spillover, which is called basically the compensation. So the compensation is calculated using the single stained controls that I have already talked about. But yeah, the controls are very important. The single stained controls is it will give the intensity for each, let's say you're measuring three different proteins with three different fluorophores. So for each fluorophore, you need to have a positive control to know where exactly or which filters the okay, maybe I will explain you in the next slide. It would be better. So here the compensation is just that because there is a spillover in the PE channel, and this is a false positive result. You want to compensate it to come to have it in the FITC channel. So this is done with your positive controls. So the unstained control is basically your only cells, and you need this to have a basic gating, that is you adjust your autofluorescence. And then the positive control that I was talking about is that for each fluorophore, you need to have a positive control, that there is a positive fluorescence, so that you know that where you should put the gating, so that anything above this or anything below this is actually a false positive result. So the positive controls are really important in that sense. So for each fluorophore or the number of fluorophores that you are using, you should have a positive control. And by positive control, I mean actual positive control, because people they tend to use, let's say, a part of your sample which might show 40 to 50% of positive control, and then they use it as a positive control, which is not correct. You should actually use positive control. It might be with a different stimulation, such as the PMA for ERK, and to have to actually plot the gating. So yes, you have to identify that for each protein, which is the positive control or which is the stimulant that I can use to get a positive signal for that particular phosphoprotein. And then there is also the FMO control, which is the fluorescence -1 control. And this is basically all of your fluorophores except for one. So this is so if in this case you see that you have three fluorophores, so here FMO control is two of the fluorophores except for one. So this helps in determining the relative contribution from overlap and correcting the spectral overlap. So if you don't have the PE fluorophore here, this means that the PE channel should not show any signal. But here, if you see, there is a signal. So this means that it's a false positive result and you need to do the compensation. So finally, I would talk a bit about the points which you consider. I have already covered a few of the points, but this is the final list of the points that you should keep in mind before proceeding with the Phospho-Flow experiment. First is your instrument configuration. Like as I already mentioned, that it is imperative to know what is the different lasers and filters which are available in your flow cytometry. Yeah, in your flow cytometry, in your flow cytometer, and also the choice of fluorophore is very important. Like the type of fluorophores which is linked to the antibody is a factor that could be affected by both dehydration and denaturation. And some fluorophores are also smaller in size, which gives it access inside the cells easily, like the FITC, the Alexa 488, and the Alexa 647. Apart from this, the antigen density is also something which you have to keep in mind, that sometimes there is very few antigens that you want to target, and for that you would need a brighter fluorophore or a brighter dye as compared to the ones where the antigen density is quite high. You can go with the dim fluorophore. And of course, this depends on the reagent availability. Not always the things that you are planning might be available in the market. So here you have to thoroughly check of what are the different vendors who are providing the different antibodies with your uh, interest fluorophore. And the other thing is the spectral overlap. So as the number of measured fluorochromes per sample increases, so the complexity of the signal which is gathered at each detector also increases. And for the multicolor panel compensation, we have to use the multicolor panel compensation to remove the spillover, because after a certain point it's difficult to do it manually. So you need these panel compensations which are already available to remove the spillover, to guarantee only pure fluorochrome signals which are associated with each primary detector. And finally, the use of appropriate controls, because without the controls, the compensations will not be successful. And based on all of these, you have to design your panel, which will make sure that there is a significant difference between the different clusters. And because if that's not the case, then if there is an overlap, then of course the signals are mixed, and then you will get a false positive signals. So here are a few of the tools, online panel designing tools that you can look at, which can help you guide with your panel design. The first one is from the BD Bioscience, and the second one is a more general one, the Fluorofinder. So yeah, it will give you a basic idea of how you should go ahead with your panel design. But I would suggest that you should always consult your flow cytometer operator to talk about talk about the practicalities of performing the flow cytometry experiment. So with that, I would like to end this talk, and thank you everyone for attending. And yeah, I would be happy to take any questions.
Thank you so much, Sharmila. Wonderful, wonderful talk. You should definitely take a look at the webinar chat. Lots of people are very appreciative of your style of teaching. So we definitely really appreciate it. You're a great presenter, and I definitely learned a lot from it. We will get to the Q&A, but before we do that, I just wanted to share just a few more slides. All right. So I just want to share just a few more slides on some of Protein Tech's offerings for flow cytometry before we begin the Q&A. In addition to our polyclonal and monoclonal antibodies, Protein Tech also offers a wide selection of recombinant antibodies that are validated and optimized for flow cytometry. All our recombinant antibody clones are carefully selected after multiple rounds of validation to identify clones with the best signal intensity and the lowest background. This includes antibodies from top-sited clones that are produced with rabbit and mouse host isotypes. So you still get the epitope and binding affinity of gold standard clones, but on the recombinant backbone that fits your panel. Our R&D team has also genetically engineered new recombinant antibody clones that have optimal performance in flow cytometry, as well as antibodies that have a mutated FC receptor binding domain, which eliminates the need for FC receptor block. And these are known as our FC0 RAB antibodies, and I'll talk a little bit more about those on the next slide. And then finally, we also have offer nanobodies for flow cytometry for immuno-oncology targets. And since all these products are recombinantly manufactured and they rely on cloning into high yield expression vectors, you get all the benefits of recombinants, such as consistent performance, high lot to lot consistency, and unlimited supply. So just to talk a little bit more about our FC0 Rab antibodies. So these antibodies are the newest addition to our flow portfolio, and they were designed to eliminate background due to FC receptor binding. So many immune cells, especially monocytes, express FC receptors, and unfortunately, the antibodies that you use for your flow cytometry experiments have FC receptor binding sites. Therefore, your antibody might be binding to your protein of interest, but it also might be binding to FC receptors and causing a false positive signal. So, to get around this, we generally use an FC receptor block to mask these binding sites from your antibodies. However, FC receptor block not only lengthens your protocol, but it also doesn't completely get rid of that false signal. And you can see that really well here in this data. So, here we're looking at CD3 and CD4 expression. Um, so you can see your cell population that is CD3, CD4 positive, and then right below it is another population that expresses CD3 and CD4, but slightly lower. And so this is going to be your kind of background. These are going to be your cells that your antibody has bound to those FC receptors. So what we're going to do is we add our FC receptor block, and you can see that you can get rid of that background just a little bit. But if you use the FC0 Rab antibodies, you can see that that background is virtually gone. So basically, what this does is this is because the FC region, the FC receptor binding site of these antibodies are mutated, so they will no longer bind to those FC receptors. And so not only do they not bind to those FC receptors, but now you also don't need to add FC receptor block, which also shortens your protocol. Additionally, these antibodies also use AccuBrite site-specific stoichiometric conjugation so that we are in control of the degree of labeling, the brightness, um, and we also eliminate the possibility that these fluorophores might be binding to your antigen binding site. Also, all our FC0 Rab antibodies use a rabbit IgG backbone so that one isotype control is needed for your experiments, and they are combined with top-sighted and reliable clones. I want to talk a little bit more about our dyes as well. So our antibodies for flow cytometry are conjugated to either conventional dyes or our in-house Corelight Plus dyes. So our Corelight dyes are bright fluorescent dyes which offer high photostability and minimal fluorescent bleed through, which allows you to use several of these dyes in a single panel. These dyes are equivalent in spectral profile to Alexa Fluor dyes, and they undergo stringent validation to ensure reliable results. In addition, we also offer antibodies that are conjugated to conventional dyes, such as FITC, PE, APC, and various other dyes as well. So you can see the list of the dyes that we have over here, and this list incorporates our Corelight dyes and additional dyes such as Atlantic Blue, which is spectrally similar to Pacific Blue, or Cardinal Red, which is spectrally similar to Texas Red. I also want to highlight really quickly our flexible antibody labeling kits. In an ideal world, when you're using a antibody for flow cytometry, it will be conjugated. But unfortunately, a pre-conjugated primary antibody is not always commercially available, or the ones that are out there just don't fit your panel. And in this situation, you might find yourself needing to conjugate your own antibody. So Protein Tech offers a quick and easy solution for this with our flexible antibody labeling kits. So Flexible uses a high affinity linker that allows you to conjugate your own antibodies in just 10 minutes, regardless of the antibody supplier or antibody buffer composition. And the way that it works is that you basically take your primary antibody and then you pipette in your Flex linker. And your Flex linker is going to have fluorophores that are covalently attached to it. And then within minutes, your Flex linker will bind to the FC portion of your primary antibody. And then what you do is you add in your Flex quencher, which we show here in purple, and that Flex quencher is going to bind to any of the unbound Flex linker, and that's it. You now have a conjugated antibody that is ready to use. And because it is really simple and fast to do, Flexible allows you to easily rearrange your panel for your multiplex experiments. Also, if you are just starting out with your panel design, Protein Tech also offers pre-optimized flow cytometry panels. These panels, which are listed right over here, they were curated and optimized by our R&D scientists to detect specific subpopulations of cells. So each population or each panel incorporates top-sided clones and is designed to have this PE channel open. And that way you can easily incorporate your target of interest into your panel. And then finally, Protitech also offers supporting resources, reagents, and kits for flow cytometry, including isotype controls, secondary antibodies, viability dyes, cell health assays, and fix and perm buffers. We also have a wide variety of blogs covering topics from panel design, gating, and data analysis, as well as protocol guides and past webinars from internal and external speakers. I also just want to mention that as a attendee of today's webinar, you will be receiving 2 follow-up emails. The first follow-up email will have a recording of this webinar as well as the other 2 webinars that happened in this series this week, and a link so you can request a free sample of one of our FC0 RAB antibodies. This is going to be the 25 test size from this selected list right here. If you are eager to do this now, you can also scan the QR code here and place your free sample request. Otherwise, do watch out for that follow-up email that will also have that link. And then you will also receive a certificate of attendance for this workshop. We do ask that you please allow 1 week for these follow-up emails to be sent. So with that, I think we can go ahead and start the Q&A. If you do have any questions after the Q&A is over, you can always email us at events@ptglab.com, or you can email Sharmila directly at sharmilab03@gmail.com if you have any questions that we don't get to today. And then if you liked today's event and want to know about all of our upcoming webinars, please do follow us on social media because we post about all our upcoming events there. So with that, I think we can go ahead and start the Q&A. So there were a few questions that there were a few repeats of, so I just wanted to start with those, if that's
Yep.
okay. So the first question is, do you do the surface staining first before you do phosphostaining?
I think it's better to maybe what you can do is you can split some of the cells and do surface staining in one of them and do phosphostaining in the other part. But if you want to do it at one go, you can. But then, yeah, you have to make sure that the staining protocols it doesn't affect the antibodies and also the spectra, the emission spectra that you are using for the fluorophores. It's quite far apart. So, yeah.
Right. Thank you. The next question that a few people had asked was, what percentage formaldehyde is used for fixation?
So usually it's said that 4% of formaldehyde, paraformaldehyde is used, but it basically works with most of the intracellular proteins. But yeah, I would say that different proteins or different cells might require different percentage. So you should always do a test run before going ahead with your big experiment, because yeah, sometimes it might happen that yeah, you are following the protocol, but then you don't get a positive signal, and then you just think that maybe your cells did phosphorylate or something, but sometimes that's not the case. So I would say, yeah, do a pilot run before you go ahead and do the big experiment that you're planning. But yeah, 4% is usually what it's recommended.
Great. Thank you so much. Before we move on to the next question, I just want to make a comment on how amazing it is to see everyone answering everyone else's questions. It's really beautiful to see, the community
Yeah,
coming together and
really.
collaborating like that. But moving on to the next question. So there was a question asking if it's ever preferable to stain cells overnight.
Uh, so ideally, I don't think it's required. Uh, but as I said, that sometimes it might happen that, yeah, there is non-availability of the flow cytometer machine or something, and yeah, you have to preserve it, then you can. But I think there will be quenching happening, and then you will lose out some of the signals. But so I won't recommend that. But if yeah, that's the situation, then maybe you can test it. But yeah, I won't recommend it.
Okay, thank you. Um, oh, this is a really interesting question. So this person's saying, I'm working on a receptor that currently has no reported intracellular signaling activity, so I cannot use antibodies against specific phosphoproteins. Do generic anti-phosphatyrosine antibodies like PY20 work well in phosphoflow?
Uh, Yep, I'm afraid I'm the correct person to answer this because I have never used that. So yeah, I cannot comment on that. Maybe you can reach out to someone who have used it. I'm not the right person to answer this question.
Okay. Yeah,
Sorry about
sometimes
that.
the specific, if you haven't done it yourself,
Yeah, so
is
I
really
don't want to
difficult to
give
answer.
like uh, I don't want to give some information which might be not correct, and then yeah, yeah,
Yeah. Yep.
it's just that yeah, it's better to ask someone who has already done such kind of experiments.
Yeah, absolutely. So then moving on to the next question that we have here. Uh, so is the positive control, when you were talking about controls, is that the same as using beads?
Yeah, so this is a nice question. So the thing is, ideally, you should use the cells that you are using for your experiment, but in the case when there might be cases where you don't have a positive you cannot have a positive control because there is no stimulation which will give you a positive phospho level. So in that case the beads help, and but I would say like I would recommend that only in the absence of the positive control you should go ahead with the beads, but if not then maybe try with different stimulants which will have a positive effect on the phosphorylation status of the protein that you are looking into.
Great, thank you. So I think we have time for maybe just a couple more questions. I know we're a little over, but I want to make sure we just get through a few more. So this next one is asking, how transient are the phosphorylation events? So do we need to fix immediately, or can we surface stain at room temp and then fix to look for phosphorylated proteins?
Uh, uh, so if I understood the question correctly, so it really depends on which phosphoprotein you are targeting because yeah, different phosphoproteins have different expression profile. So for each there are certain phosphoproteins which are very quick like the nuclear proteins, they are very quick, so you have to be and it's actually very challenging or tricky to measure the nuclear phosphoproteins, but many phosphoproteins, yeah, you can check the dynamic levels because they usually the ERK or the AKT, they have the maximum response at 15 minutes, but then people also check at 5 minutes and 10 minutes or yeah, even 1 minute. So it basically depends on what you are looking into or what kind of phosphorylation level you want, or for what is your main uh, how to say main aim. So yeah, I think it depends on that. So yes,
Yeah, so maybe the first step is like doing a time course and determining how long can
uh,
you see
yes, or
your
else there are
signal.
a lot of resources available, so you can just do a literature search and then see what people have used. Ideally, they mention the time which gives the maximum excitation or the maximum phosphorylation, but it can be specific to certain cell lines as well. So as the protein changes or the cell line changes, the phosphorylation event also changes. So yeah, keep in mind that before you can decide on yeah if you want to measure protein at 5 minutes or 15 minutes. Yep.
Great, thanks. So I think maybe a couple more questions. So the next question is asking, can I use a PE fluorophore to detect two markers at the same time and emit two different colors by using different filters?
Yes, absolutely. Laser is it's just the excitation which provides the laser is for the excitation. But if there is different emission spectra with the different filters, you can definitely measure the different proteins that you are targeting.
Great, thank you. All right, so let's do this last question. So for this last question, they're asking if I want to keep the cells alive and avoid using formaldehyde, how can I measure cytokines or other signals involved in specific activated signaling pathways?
Uh, uh, yeah, okay. If you don't want to use formaldehyde, so you if I may ask, do you want to like check the signaling profile over time, or at a particular? Because if you want to do it at one time, then you have to fix the cells. Otherwise, the phosphorylation can keep on going, or it might also get dephosphorylated. So I would say that if you want at a particular time, then yes, you have to do the fixation. But if you want to look at the phosphorylation level over time, then I think, yeah, for fixing the cells is not required. You can do live cell imaging to check the phosphorylation level.
Okay, great. Thank you so much. Yeah, since we're kind of getting 15 minutes over, I think we can end it there. I know we didn't get to all the questions, and I really
Yeah, you can
apologize for
always
that.
email
But
to me
yes,
your personal
that's
questions.
exactly what I was going to say. Feel free to email. I'm going to put up the emails one more time on the screen so everyone can see. Yeah, feel free to email Sharmila, or you can email us at events@ptglab.com as well, and we can always forward it over to her. But I just wanted to thank you once again, Sharmila, for the wonderful presentation. And again, I know this is your Friday evening, so we really appreciate you spending the time to teach us. And I just want to thank everyone who joined today as well. Thank you for taking the time to kind of learn a little bit more about Phospho-Flow. Hopefully, you guys all found it useful. And with that, I think I'll go ahead and end the webinar. And I just want to say have a good morning, afternoon, or evening, depending on where you're tuning in from the world. So take care, everybody.
Yeah, thank you so much.